All right, this is going to be the third and final lecture video for Chapter 6. So we left off with finishing talking about Phase 2, and now it's time to talk about Phase 3, the electron transport chain. In other words, what we do with all of that NADH and that 2-FADH2. So at this point, we liberated 2 NADH during glycolysis. We liberated 2 NADH during pyruvate oxidation. And then in the electron then during the Krebs cycle, we liberated 6 more NADH. So that is 10 NADH plus hydrogen ions. So 10 hydrogen ions, 10 NADH, all rolling into the electron transport chain along with that 2-FADH2. So it's time to talk about what happens. The electron transport chain is called a chain because it is a series or chain of membrane proteins. These membrane proteins are embedded in the inner membrane of a mitochondria or the straight cell membrane of a prokaryote. But prokaryotes, who cares? So each one of the membrane proteins is an electron acceptor, an electron carrier. In other words, each one of those membrane proteins is going to bind to an electron. When you gain an electron, that's reduction. So we're still carrying out oxidation reduction reactions. So NADH2 or NADH plus hydrogen ions brings 2 electrons and 2 hydrogen ions to the beginning of the electron transport chain. At the very end of the electron transport chain, we've got oxygen. So let's see how this works. Oxygen is going to accept electrons and hydrogens in order to make water. So 2 hydrogens and 2 electrons are 2H. 2H plus half O2 is H2O. So O2 being at the end and binding 2 electrons makes it the final electron acceptor in cellular respiration. So let's take a good look at it. NADH is going to release its electrons and hydrogen ions into the chain. And the electrons are going to move from protein to protein. Whereas the hydrogen ions are going to be pumped out of the mitochondrial inner membrane and into the intermembrane space, the space between the mitochondrial outer membrane and the mitochondrial inner membrane, intermembrane space. So you're essentially cranking a bunch of hydrogens into the intermembrane space. So let's take a look at NADH and how it's able to separate electrons from hydrogens. So when we talk about NADH, it's an NAD, which is this black rectangle, bonded to a hydrogen. And we don't put H plus there because even though right over here, this is bonded to the proton, and this is the electron, we've still got a proton and an electron, so we've got one functional hydrogen here. It's just the proton and the electron are carried separately on NADH. So, good to know. And then this over here is a hydrogen ion because its electron is right here. So the 2 electrons from the 2 hydrogens associated with NAD are carried separately from the hydrogen. That's why this H plus that isn't directly bound is an H plus. Its electron is over on the NADH. Okay, so what we're establishing here is that NADH can release electrons separately from hydrogens. And when it releases the electrons, then the hydrogens being released are going to be singular protons or hydrogen ions, H plus, because an H plus is a single proton. Okay, so here we go. Step 1 in the electron transport chain. NADH rolls up and releases its hydrogens and electrons. and that converts it back to NAD. And now NAD plus can roll back up to phase 1 and phase 2 in order to bind some more hydrogens and come all the way back to the electron transport chain. So it's a big loop. So it's going to release the electrons separate from the hydrogen ions. The hydrogen ions enter in here and they're pumped out. At the same time, some of these hydrogen ions don't go directly into the first membrane protein, and so they're pumped out of this membrane protein or this membrane protein. They can be pumped out of any of these first 3 membrane proteins. Not that you need to know that it's any of the first 3 membrane proteins. What you need to know is that hydrogen is pumped out of the inner membrane. This is the intermembrane space. The electrons are passed from protein to protein. So this protein here is going to oxidize this protein and become reduced. So each protein is reduced until we've got all these electrons finishing up, and over here you see 2H plus half O2 yields H2O. So these are our hydrogen ions here, 2H plus half O2. And we can't really have water that has, cations in it, so the electrons are passed right into here during this process in order to make some nice, stable, regular old H2O. So the electrons are half the picture in producing H2O. Let's talk about where these hydrogen ions come from. Well, right over here. When we pump hydrogen ions into the intermembrane space, this creates a concentration gradient. The hydrogens are more concentrated on the outside of the cell, or on the outside of the mitochondria, than they are on the inside. And thus, they're going to follow the rules of diffusion. Hydrogen is going to diffuse back into the interior of the mitochondria, into the mitochondrial matrix. Now, it is the diffusion of hydrogen across this protein which actually allows us to convert adenosine diphosphate and a spare phosphate into ATP. So, hitting it by text. So, hydrogen in the form of hydrogen ions is pumped into the intermembrane space. Puts more hydrogen outside the inner membrane than inside, and so they're going to want to diffuse from areas of high concentration to areas of low concentration, from the intermembrane space into the inner membrane. The enzyme that makes ATP pumps the hydrogen back into the mitochondria according to the rules of diffusion, which makes it passive transport, which means it doesn't require energy, which is good because this is what's making the energy in the first place. It'd be kind of lame if we had to spend energy to make energy. That enzyme is called ATP synthase. Synthesis, making the enzyme that makes ATP. So like I said, the movement of hydrogen is what drives phosphorylation of adenosine diphosphate, ADP. Phosphorylation is my fancy science term for adding a phosphate onto ADP and turning it into ATP. So when they arrive back inside the inner membrane, back inside the mitochondrial matrix, those hydrogen ions are combined with the electrons and oxygen in order to make water. So 2 hydrogen plus half O2 yields H2O. And there it is again. Hydrogens pumped into the intermembrane space, which creates a diffusion force, which causes the hydrogen to go through ATP synthase back into the mitochondrial matrix. At the same time, the electrons move protein to protein until they get to here, where they combine with the hydrogen and the oxygen to make water. And the movement of the hydrogen across this protein is what causes ATP production. It's what allows ADP to combine with phosphates to make ATP. Now, we talked about in glycolysis, the production of ATP is substrate level phosphorylation. Well, in the electron transport chain, oxygen is the final electron acceptor, so we call it oxidative phosphorylation. So ATP is produced by hydrogens moving across the mitochondrial membrane and driving ATP synthase. Let's take a look, shall we? Maybe, perhaps, if we're lucky. Okay, this is a good video.
ATP synthase is a molecular machine.
You're not going to need to know really any of the text here. So in fact, I'm going to mute it and just sort of give you the basics of what's going on here. So right here, these little animations coming in are ADP and phosphate. And when the red leaves, that's ADP and phosphate being converted into ATP. So what's cool about ATP synthase is that it's a rotary pump. It's this mechanical pump that rotates. So it's actually got a rotating part to it. So hydrogens enter in and bind to that rotating part, and the binding of the hydrogen is what causes the protein to change shape. In this case, that shape change is represented by rotation. So there it is, rotating. Pretty cool. So this says protons. Remember, a hydrogen ion is a proton. So there we go. They're moving through, and then as it rotates full circles around, hydrogen comes out into the mitochondrial matrix. So let's take a quick look at what exactly is going on here. So let's take a look from above. So every time this rotates, it pushes the molecule around. So let's take a look at how that rotation works. All right, shall we? So it's going to rotate here, and when it pushes it around, this pulls in and allows ADP and phosphate to bind, and then when it rotates, they're created or bound together to make ATP, and then it pushes them out. So that's sort of the gist of it. You don't need to know the details of how ATP synthase works. I just think it's really cool to know that it is actually a rotor. And because this is passive transport, by the way, this will work backwards. So when there's too much ATP or too much hydrogen ions inside the cell, this thing will just flow backwards and convert ATP to ADP plus P. Just kind of neat. All right, back to the slideshow. So the final tally of phase 3. The electron transport chain alone makes 29 to 34 ATP. It's a bunch. That is not too shabby, considering glycolysis only produced a net of what? That's right, 2 ATP. It only produced a net of 2 ATP. So let's add our ATP in there. So glycolysis created 2 ATP. Krebs cycle created 2 ATP. Then the electron transport chain made 29 to 34. So that is roughly 33 to 38 ATP for all of cellular respiration. Pretty darn efficient. That oxidative phosphorylation, that electron transport chain, is the single most efficient producer of ATP in all of cellular respiration. And that is exactly why every single one of our cells uses oxygen in ATP production. How about cells that don't use oxygen? For instance, cells buried deep in the soil aren't really exposed to oxygen. They're in anaerobic conditions. Well, how do they work? One of two ways. The first is anaerobic respiration. And in this phrase here, respiration is an important word. Okay? Because this is anaerobic respiration, we are actually still using an electron transport chain. And we're still using the Krebs cycle. However, oxygen is not the final electron acceptor. That's right, oxygen isn't the only possible final electron acceptor. In fact, any heavily electronegative molecule will do. So, other organisms might use nitrate, which has nitrogen, which is electronegative, highly electronegative, or they may use sulfate, which has sulfur, which is another electronegative atom. And then my favorite category, other. So detailed of me. So detailed. Anyway, this is still cellular respiration. We're still taking glucose and breaking it down. However, it won't be able to go through every single step of the Krebs cycles. A couple of those steps in there are oxygen dependent. You don't need to know which ones. Just know that we're not going to be able to go through all the steps of anaerobic respiration. So we're not going to get as much in that electron transport chain, as much NADH in the electron transport chain. So we're going to have slightly less energy production, but still a large amount compared to the energy produced in glycolysis. So this is anaerobic, which means it requires, or it does not require oxygen. Okay, how about the other way to be anaerobic? Well, fermentation. So fermentation is anaerobic, and it only produces two ATP. And in fact, those 2 ATP are produced directly by glycolysis. The only ATP producing step in fermentation is glycolysis. There are 2 basic types of fermentation. Alcoholic fermentation, where we have glycolysis occurring, converting glucose to pyruvate, and then pyruvate being converted into ethanol and to carbon dioxide. This is the basic kind of fermentation produced by yeasts that are used in both bread. Carbon dioxide production is why bread dough rises. It's gas bubbles forming. And in beer production, ethanol. That's the beer we drink. All right. So the products are a net 2 ATP, and the byproducts are 2 ethanol and 2 carbon dioxide. As you can tell, me specifying that alcoholic fermentation has the products of 2 ATP and 2 ethanol and 2 CO2, pretty important. Remember, when I write 2 ATP here, this is 2 net ATP, because this was glycolysis. So, like I said, alcoholic fermentation involved in making beer also happens to be a regular function of a lot of yeasts or other microorganisms. All right, now it's time for the other kind of fermentation, and that's lactic acid fermentation. So lactic acid is the production of lactate, which is an acidic molecule, so hence lactic acid. In this case, glycolysis occurs and converts glucose to pyruvate. Big surprise there. And then pyruvate is converted into lactic acid, or lactate. The products of lactic acid fermentation are, amazingly enough, 2 ATP. And then the byproducts are 2 lactate, or 2 lactic acid. Fermentation actually occurs in your body, and it happens in your muscles. In fact, your muscles do not require oxygen in order to stay alive. They don't do it very well, but I think it's kind of useful to have, living skeletal muscles. So, when your muscles are under stress, when you're exercising a lot, like running from a bear for some of you, or, for me, making the 2-day trek to the refrigerator from the couch, have to camp about halfway there overnight to regain my energy. Your muscles use up all of your oxygen, and at a certain point, your breathing rate is as fast as it's going to get, and you're not going to get enough oxygen to actively supply every single one of your muscle cells with the oxygen it needs. Since those muscle cells are still doing work and they don't have oxygen around to make the ATP they need to do that work, your muscle cells are going to switch to fermentation, which is pretty cool. By the way, lactic acid is thought to be one of the sources of the burn. Feel the burn of the exercise, right? Part of that burn is thought to be due to lactic acid. Another part thought to be due to little micro tears that occur in your muscle whenever you heavily use them. Okay, so little bit of vocab. Obligate, anaerobe. When you are obligated to do something, what does that mean? Right? My mom's throwing a party. I'm obligated to go. That's right. I have to go. Obligate anaerobes have to use anaerobic respiration. There's no way around it. They absolutely have to use anaerobic respiration. In fact, if you expose an obligate anaerobe to oxygen, chances are the oxygen is just going to straight kill it. So, obligate anaerobes can only undergo fermentation, or they can only undergo anaerobic respiration. They cannot switch between either one, so either a species is fermentative or it uses anaerobic respiration. There's no switching, unlike muscle cells, which can switch between aerobic and fermentation. Obligate anaerobes only get to choose one. So let's contrast obligate anaerobes with facultative anaerobes. Facultative, that's a fancy schmancy word for doesn't have to do it. So these are cells that can undergo fermentation and aerobic respiration. Notice I did not say aerobic and anaerobic respiration. That's because if you have aerobic respiration, your final electron is oxygen. Anaerobic respiration does not have oxygen as the final electron acceptor. So, mutually exclusive. So, facultative anaerobes are always using aerobic respiration unless their oxygen content dries up. Unless the amount of oxygen they have, unless their source dries up because you're exercising more than you can breathe enough oxygen to supply your muscle cells, once that oxygen dries up, switches to fermentation. So, aerobic when oxygen is available, anaerobic when oxygen is not available. Okay, one final note on evolution. The oldest prokaryote fossils are something like 3.5 billion years ago. Now, we do not have fossilized single cells, but these old prokaryotes sort of formed colonies, and they produced these big, thick hunks that look like rocks because, well, they're fossils. They are rocks. But they're called stromatolites, and they're these big sort of bulbous ocean rock-looking things sticking out of the water. Those stromatolites are some of the oldest fossils. They were produced by prokaryotes roughly 3.5 billion years ago, only 500 million years after the Earth was formed. So, when was there enough atmospheric oxygen for aerobic respiration? 2.7 billion years ago. Okay, so that leaves 8 800 million years in which there wasn't enough oxygen for aerobic respiration. What the heck were they doing for 800 million years if they weren't able to use oxygen? Well, the answer may shock and amaze you. Glycolysis. It is thought that glycolysis was the very first form of energy production seen in the biological world, in any kind of cell. And this is a pretty safe assumption because if it's alive, it's undergoing glycolysis, except for the viruses, but nobody likes them, so we're leaving them alone. So if it's alive, it's undergoing glycolysis. Since glycolysis is shared by every single living thing, that's pretty good evidence that it was one of the very first cellular processes. The older the cell, the more likely we are to have some kind of features associated with it. So the fact that that feature is in every single living cell, no matter the species, is pretty decent evidence that glycolysis was the very first energy producing pathway. And for 800 million years, it was all those organisms needed. But atmospheric oxygen increased 2.7 billion years ago, at which point organisms started to, well, either they went extinct because of that oxygen poisoning the atmosphere, or they adapted and were able to start up some aerobic respiration. So that's just pretty darn cool. So glycolysis is one of those pieces of evolution that connect down through every single living cell. I love that. It's very cool. Affirms the fact that all life is related, and so all life is family. That's pretty neat. So that's my closing thoughts. All of life is family. All of life uses glycolysis, except for lame viruses. Viruses actually aren't lame. They're pretty darn awesome. But I don't care about them right now. That is it, I think. Let me just check, but pretty sure this is our last slide of Chapter 6. So, take a rest. Take a break. Process it. See you guys later.